Hello, everyone. I'm Stuart Diaz, account manager for the Northeast Biopharma um, segment at CST. And I want to welcome you today to the webinar on customized proteomic solutions for expedited drug discovery. I would like to introduce Dr. Matt Stokes, who is Associate Director of Proteomics Products and Services at Cell Signaling Technology. Matt provides start-to-finish proteomics solutions for both academic and industry clients. Today, we will describe the application of proteomic assays and data analysis to the challenges of early to mid-stage drug discovery and development. During both live sessions, there's two, you'll have the opportunity to ask questions by typing them into the GoToWebinar questions area in your control panel. So without further ado, I'm going to hand it over to our proteomics expert, Matt Stokes.
Hi, everyone. This is Matt. Thanks for joining us today. I'm really excited to be able to talk to you a little bit about the work that we do in mass spec-based proteomics. But first, I wanted to start with just a few words about CST in general. So as a company, we were founded in 1999. And since that time, we've been developing and manufacturing antibodies in our Massachusetts facilities. We're a company that tries to have an environmental focus, be active in our community, and also be a charitable contributor. And most importantly, I think, we really are a quality-driven company of scientists for scientists. And it really starts at the top. So our CEO, before founding the company, was a PI in an academic lab. So right from the top, I think we have a pretty unique understanding of the challenges that bench scientists face in the lab every day. And in my role as Associate Director of Proteomics, a question that I kind of unfortunately get a lot is, wait, you do proteomics? Everybody knows CST is an antibody company, but yeah, not only do we do proteomics, but we were actually kind of one of the early adopters of mass spec-based proteomics. So we pioneered what we call PTM scan, which is combining antibody enrichments with mass spec all the way back in 2003. This is one of the first papers that came out in 2005 looking at tyrosine phosphorylation. And we started doing client services in 2004. And since that time, we've done over 1,500 services and been involved in hundreds of publications. But yes, we're an antibody company, so it all did start with antibodies and this kind of unique class of antibodies that we developed. So typically when we're going to make a new antibody, we use a single peptide or part of a protein or even full-length protein as the antigen. Here we're showing a phosphopeptide. So we raise antibodies against that peptide, purify, and get something that's nice and specific for a single protein site. Now, this new class of antibodies that we made were a little bit different. So instead of a single sequence as the antigen, we actually started using degenerate peptide libraries. So again, here we have a phosphopeptide, phosphoserine, and we have an arginine at -3. And so those two residues are fixed and they're present in every peptide in that library. And then we vary all the other amino acids around it. And what we get when we do this are antibodies that are really exquisitely specific for that targeted motif, in this case, RXX phospho-S, but will broadly react with a lot of different primary amino acid sequences. So we can do this for phosphorylation motifs. We can also do it for other PTMs like I'm showing here with acetylated lysine, again, using that degenerate peptide library approach. So we made these antibodies and we used them in Western and IP Western, even some other applications, and they were working great. But with the advent of mass spec-based proteomics, we thought maybe we could start using these as the enrichment tools in front of doing a mass spec experiment. And again, that's what we call PTM scan. And so you take your material, digestive peptides, so these are peptide level enrichments, enrich with one of those PTM or motif antibodies, run mass spec, get your peptide identifications, and then do quantification to say what peptides, what proteins or sites are up or down across the different samples that you run. And for us, it kind of all started with tyrosine. That was one of the first PTM or motif antibodies that we made and really got working well along with the mass spec. So we went a little bit crazy and we profiled basically any sample that we could get our hands on with phosphotyrosine, including a number of cancer samples, both cell lines and tissues. And this is a paper that came out a few years ago looking at tyrosine phosphorylation in ovarian cancer samples. And so profiled a bunch of ovarian lines with phosphotyrosine, ran the mass spec. And what we noticed was that in a subset of these, we were seeing strong phosphorylation of the receptor tyrosine kinase ALK. Did a little follow-up work and actually identified a novel gene fusion of ALK and FN1. And importantly, cells harboring that FN1 ALK fusion are really sensitive to crizotinib. So over on the left here, we're looking at tumor growth over the course of a week. The green line is vehicle control and the blue is crizotinib. And you can see that the crizotinib treatment almost completely abrogates growth. On a more molecular level, we can see this as well. So on the right, we have some Western blots with ALK and phospho-ALK antibodies. You can see strong phosphorylation of that FN1 ALK fusion in the vehicle control. And again, that's almost completely gone with crizotinib treatment. So we did this in ovarian. We also did a number of other studies in other cancer types like non-small cell lung cancer. Some of those papers are shown on the left here. And that work together led us to development of some really nice ALK antibodies, including this D5F3 clone, which works great in Western IP, or as shown here on the right, IHC. And we actually even took things a step further and partnered and came up with a companion diagnostic for ALK for lung cancer patients, a step that was called a significant step forward in the war against non-small cell lung cancer. So really, I think kind of a cool story from those early days of how we did these kind of discovery experiments, in this case with phosphotyrosine, got good, reliable data, validated that data, and then we're actually able to take that all the way through to defining disease drivers and coming up with companion diagnostics for those disease drivers. So today, we don't just do internal work. As I said, we started doing services back in 2004. And for those services, people send us in material, and it can be cell lines, it can be tissues. We've done plant material, whole organisms, microbial samples, primary cells or sorted cells, even serum or plasma. We take that material, we process those samples, we run the mass spec, and then we do data analysis and come back with really a nice finished data package that tells you, here are the proteins and here are the sites of modification that we've identified. Quantitatively, here's what's different between samples. And then we try and provide a little more value to that data by doing things like providing a summary and walkthrough that takes you through these tables and explains things. Guides to prioritizing candidates for follow-up. And then I think really importantly, after the project's done and we've delivered the data and you've had a chance to digest it a little bit, we sit down, we have a meeting, and we actually go through the data together. We have a follow-up consultation and just make sure everything's clear and really make sure that you get the most value out of that data possible. So there's kind of three main areas that we work in. We do total proteomics. And so here we're looking at unmodified peptides to tell us about changes in protein levels across your samples. And when we do total proteomics, we're usually asking for on the order of micrograms of protein per sample. We do a lot with PTMs. So here we're able to identify and quantify thousands of post-translationally modified peptides. And we'll look in a minute at all the different PTMs that we can now assay. And depending on what we're looking at here, we're generally working in the neighborhood of milligrams to protein, although for some enrichments, we can go down to micrograms of protein. And finally, we can do targeted assays, or what we're calling here IPRM proteomics. And this is a great kind of next step. So after you've done these discovery type experiments and identified a group of markers or drivers in your samples, and you really want to profile that more limited subset of endpoints across a lot of samples, hundreds of samples, thousands of samples. we can help to design, validate, and even run those targeted assays. And the input material for that is going to vary based on the endpoints you want to look at, but typically our goal is to be around 100 micrograms of input protein per condition for that. So how are people using our services? Well, we do a lot of work in drug discovery profiling, doing things like biomarker discovery, target validation, looking at mechanism of action, off-target effects. But we also do plenty in just more basic research, looking at things like disease mechanisms or overexpression or knockdown of your favorite gene, protein-protein interactions, all these different things. And it's really pretty varied, the applications for mass spec-based proteomics in a lot of different research areas and types of studies. So I wanted to talk a little bit about the total proteomics. So when we run total proteomics, we can just take the samples, digest, clean them up, and run them on the mass spec. But more often now, we're actually using a multiplexing strategy, using what's called tandem mass tags or TMT reagents. And this allows us to multiplex, in this case, up to 10 samples together. So you digest the peptides, you label them with these 10 different TMT reagents. And the cool thing about TMT is that the intact peptides, even though they all have these different TMT tags on them, they all have the same mass. So that kind of stacks up in the mass spec and it doesn't ruin your sensitivity. You would hate to be, right from the beginning, dividing your signal into 10. So it all stacks up for identification. And it's not until you actually start fragmenting those peptides that pieces of each of these tags come off, what we call the reporter ions. And each of those reporter ions has a different mass. So now you can say, okay, I have this much peptide in sample 1, this much in sample 4, this much in sample 10, etc. And we do a lot of these studies, and actually most of the studies that we do for this are more than 10 samples, more than one plex. And when that's the case, the only change that we make is that we reserve one of those channels for a reference. Usually what we do is we take a little bit of each of the samples we're going to run in the study, and we mix that together and use that as that first channel. What that allows us to do is to compare data across different plexes. We really have a nice complete data set. So we take, digest, we label with the TMT, and then we combine those samples into one unit. And now because they're combined together, we can actually fractionate. So we do a basic reverse phase fractionation, non-sequential concatenation of those samples. And this is really big for depth of coverage, right? So instead of trying to get all the information in one shot on the instrument, we're actually running 12 fractions with long gradients on the instrument. And that really helps us to dig down and get to lower abundance proteins and really get a lot of proteins identified and quantified. So from there, we'll take those fractions and we will run them on the instrument. We're using our Fusion Lumos instrument, which in addition to being very fast and very sensitive and pretty cool looking, comes preloaded with some software to be able to do this TMT quantitation in a more accurate manner. And that actually ends up being really important. So what I'm showing over here on the right is just a very simple experiment where you mix two samples at known ratios, 1:1, 4:1, and 10:1. And the red bars are just showing what the fold changes would be based on the mixing that you did. So with the old way of quantitating, it's called just an MS2 method, the 1:1 sample looks fine. But instead of a 4:1 ratio, we get about a 2:1 ratio. Instead of a 10:1 ratio, we get about a 4:1 ratio. So why is that? Well, with this old method, there's just not a lot of specificity in the quantitation. So if you have two peptides close to each other, their TMT reporter ions can actually kind of contaminate the signal of the other one. And so you end up getting this ratio compression. So the old way is really not an accurate way to do TMT quant. And having one of these newer generation instruments that comes preloaded with the software to do it the new way becomes really important. So when we do this now, MS3 or multi-notch MS3 quantification shown in the blue bars, it really makes the situation better. So 1:1 is fine. Now you've got a 4:1 change where you expect it, and essentially a 10:1 change where you expect it. So much more accurate quantitative data. So we wanted to be able to generate some sample data, and we also have been using TMT for a long time internally. And And with all the antibody development that we do, an interesting or important thing that we want to have is knowing what cell lines express what proteins. And so we took 20 cell lines that we use a lot for our regular antibody development programs and ran them through this TMT total proteome. And so over here on the right, each row in this heat map is a different protein that we've identified and quantified. Overall, over 8,000 proteins identified in this little study. And for this view, we actually just made an average signature across those 20 and then compared each individual cell line to that average signature to say, here are things that are up or down relative to the average. And then we validated some of that data with Western blot. So this is FOXA2. Up top, we have the Western, and down below, we have the TMT total proteome data in the bar chart. And so you can see really nice correlation between the mass spec data and what you get in the Western blot. Everything agrees really well. Same thing for ZAP70. So we have a couple of T cell lines in here and CD3 epsilon. So in all cases, really nice agreement. We're getting not only huge data sets doing this, but really quantitatively accurate data sets as well. So, some of the advantages of our total proteomics, internally, we were one of the earliest adopters of TMT technology. We've been doing it for 10, almost 15 years now, I guess. So we have a lot of experience with it. And more recently, we made the decision to actually take out a license to be able to do it for services. So we are fully licensed. Everything is above the board there. We have that Fusion Lumos instrument, which, as I mentioned, is very fast, sensitive, and does the accurate quantification. Over the years, we've built up a really robust software package for interrogating this kind of data. We work under defined timelines. And then we also have, as kind of a companion service, pathway analysis services available. So we use Ingenuity IPA for that. And that can be really useful for putting the data in a broader biological context. And that's available not only for the total proteomics, but really for any of the services that we do. So for a total proteome experiment, we ask for about 100 micrograms of protein. We can and we have worked with much less material when that's been necessary, but 100 micrograms is usually on the order of a couple million cells. And our turnaround times for a single 10-plex, it's about 6 weeks. 2 10-plexes is not 12 weeks. When we start adding plexes, a 2 10-plex study might be 6 to 7 weeks. A 4 10-plex study might be 6 to 9 weeks. So that's our total proteomics. As I mentioned, it really kind of all started for us with the PTMs and in particular with phosphotyrosine. And again, here we're identifying and quantifying thousands of sites of post-translational modification. And this is a list of all the different PTMs that we can look at using this technology. And I'm going to focus most of the rest of the talk on phosphorylation, but I just wanted to briefly mention a couple of different PTMs, other PTMs that I think are kind of cool little stories. And the first one of those is ubiquitination. So when you have a ubiquitinated protein, so the target protein is shown here in the middle in blue and the ubiquitin is in green here on the left. If you trypsin digest that ubiquitinated protein, you get peptides from the target protein, and then everywhere you had a ubiquitin on a lysine, you get this diglycine remnant hanging off. So we actually made an antibody against that diglycine remnant. And again, that was using that degenerate peptide library approach. And this antibody works fantastic. We identify and quantify thousands of sites of ubiquitination in cells, tissues, other materials. And that antibody is also pretty unbiased. The only real key, as shown in the sequence logo, is having that lysine with the diglycine hanging off of it. There's no other strong amino acid preference really anywhere else in the peptides that we're identifying. The other thing I wanted to talk just briefly about is methylation of arginine. So in methylation, we can look at methyl lysine, we can look at methyl arginine. And for arginine, we have 3 different antibodies. So we can look at monomethylation, and then we actually made antibodies, and we're able to get antibodies that distinguish between asymmetric and symmetric dimethyl arginine. Okay, so between these very highly related species. And first of all, I think it's pretty cool that we were able to find the clones that could differentiate between these two. This is just some ELISA data showing the specificity of the asymmetric dimethyl arginine antibody for the ADMA peptides. or the symmetric dimethylarginine antibody for the SDMA peptides. So those really work great, and they really are specific to those different modifications. So what about phosphorylation? Well, like I told you, it all started for us with tyrosine, but since that time, we've built up a big group of phosphoserine, phosphothreonine motif antibodies. And a question I get a lot is, well, you have one for tyrosine, why not have one for serine, one for threonine, and be done with it? And, there's a couple reasons why we did it this way. One is that it's very hard to make a truly context-independent phosphoserine or phosphothreonine antibody. We do a lot of validation of these, and whenever we tried to make one and validate it, what we would see is some secondary preference for another amino acid or amino acids around that site of modification. The other reason that we really like this approach is that it allows us to get depth of coverage. So each of these is looking at a more defined space within the phosphoproteome. But within that defined space, we're now able to dig down deeper and find lower abundance proteins or lower stoichiometry sites. So we can use these phosphoserine threonine motif antibodies singly. We have some premixed formats, for example, all the basophilic kinase substrates or proline directed or everybody together. And we can also make custom mixes. So if you were interested in, say, AKT and pKa, we can make a custom mix of those to do a single enrichment for a service. So we have those PTM and motif antibodies to look at phosphorylation. But another service that actually is really getting more and more popular is not to use an antibody, but to use a metal ion on a bead. It's called an IMAC enrichment. In this case, it's a coordinated iron ion. And in principle, that will pull down any phosphopeptide. The sequence logo is shown below there. So you get these really huge data sets, and you're able to get those from less input material than you might need for a typical antibody experiment. And that's kind of shown here in the Venn diagram. So the IMAC is in the middle. It's the biggest circle. You get the most data out of an IMAC enrichment. It's a great tool for when you want to go very broad and try and get the most data possible from one analysis. But the interesting thing that we've found and others have found as well is that you see kind of complementary sets of peptides identified depending on how you enrich. So that's true for these different serine threonine motifs here in the IMAC. The overlaps can be pretty low. And I think it's especially critical for tyrosine. So you really don't get great coverage of tyrosine with the IMAC. The tyrosines are lower abundance. So you really need that more specific enrichment in order to get good coverage of the tyrosine phosphoproteome. Now, maybe to complicate things a bit further, we also have another way that we look at phosphorylation, and that's a pathway-based approach. So here the method is the same. We are digesting the peptides, enriching with antibody, and running mass spec. Only difference are the antibodies that we're using. So for this pathway approach, we actually have made multiplex cocktails of our site-specific antibodies, our catalog products. We can make a mix that has AKT473 and ERK activation loop sites and PI3 kinase and ATM or ATR. And we've made a bunch of different versions of these pathway-based enrichments. The most popular by far has been the multi-pathway enrichment, where we took all these different signaling pathways and said, okay, what are really the critical signaling nodes for all of these different signaling spaces? And let's make one reagent that with a single enrichment, we can target all of these different signaling areas and read out what's happening to those critical signaling nodes. And this really works great. We see a lot of peptides and phosphopeptides identified. And it's really pretty heavily kinase focused. So about 30% of the data that we get back are from kinases. Not surprising given the kind of central importance of kinases in all these different signaling areas. Now, unlike a motif antibody or an IMAC enrichment, where that will work in essentially any species that has phosphorylation, because these are site-specific antibodies, they've specifically been validated for human and mouse. We have actually done a few studies in other species, and it tends to work pretty well, I think, just because of the conservation that you tend to get of amino acid sequence around these important sites of modification. But primarily, it's designed for human and mouse. And overall, we know we can see about 4,000 different sites of phosphorylation on about 800 different proteins. Now, not all of those things are going to be expressed or phosphorylated in a given cell line or even a given tissue. But we know through our years of validation that this is kind of the universe of things that we can see with this reagent. We have that list available online. So when would you use these different things? Well, I like to tell people the PTM or the motif antibodies are great for depth of coverage, like we talked about. It allows you to get down to lower abundance things, lower stoichiometry things, really important for tyrosine. IMAC is great for breadth of coverage. So if you want to take one shot, do one enrichment, and get the broadest survey possible and really the most data possible, then IMAC is the right choice. And the pathway-based enrichments are great for more targeted coverage, and not really targeted in the sense of how we're running the instrument, but targeted in the sense of we're not trying to discover new biology here and find new sites on new proteins. We're really just interrogating what's happening with known pathways and proteins and sites that we already understand to be important in some aspect of signaling. And I will say a lot of people actually pick combinations of these. So for example, doing phosphotyrosine with the motif antibody and IMAC to cover serine threonine and tyrosine, or doing the IMAC and a pathway-based enrichment, kind of looking at and getting both sides of the coin and getting the more specific data on known things with the pathways, but then the broad discovery data with the IMAC. So sometimes people come to us and they know exactly what they want to do. We want to look at ubiquitination, we want to look at acetylation, phosphotyrosine. And in those cases, it's great. We just start the study. But other times people come and say, well, I have a compound, it's giving an interesting phenotype, but I really have no idea how that's being mediated. And now we have, 30 or 40 different tools to enrich and look at different PTMs on those samples. And that really becomes an untractable problem. So what we did was we came up with what we call kinome view, which is just a Western blot prescreen using those same PTM or motif antibodies that we would use for the mass spec enrichment. And if we see changes in the Western blots where we're looking at 20 to 50 species on the membrane, we know we're going to get data, really great data, when we start looking at hundreds or thousands of things in the mass spec assay. Kinome view is also a great chance to refine experimental conditions, picking things like the right cell line or dose or time point, etc. So this is some sample data. Each of these panels is a blot with a different PTM or motif antibody. We can zoom in on one. This is the PK substrate motif, phosphoserine threonine with arginine or lysine at -2 and -3. And just seeing the changes in band patterns, band intensities across these different samples, we know this would be a great tool to use for enrichment in front of mass spec. And then if we were using this also to refine experimental conditions, we'd say, okay, here's a control for cell line B in lane 7. And the biggest difference we see is with this treatment here in lane 11. So if we were picking a binary comparison, we would recommend looking at these two samples. So we kind of put all this together to give some examples of what the data looks like when you're using these different methods for profiling phosphorylation. And a pretty simple experiment. So we took some Jurkat cells, untreated, or did a CD3, CD28 stim, digested to peptides, and then did these 3 different types of enrichments. We did the pathway-based enrichment with the multi-pathway, a motif antibody enrichment with phosphotyrosine, and then the IMAC enrichment. So we take that, we run the mass spec for each of those, get our identifications, and then do quantification to see what's different between the minus+ stim samples. And qualitatively, like we saw before, in terms of just bulk number of identifications, IMAC wins. It's the biggest circle here, the most things identified. But really look at the overlap, or really lack thereof, between these different enrichment methods. So 99% of the unique phosphopeptides we identify were only found in one of the 3 enrichments. So it really points to the complementary nature of these different studies, these different assays. We can look a little more specifically at the T-cell receptor signaling pathway. And in yellow here, we're just lighting up those proteins for which we can see phosphopeptides using at least one of these enrichments. And overall, the coverage is pretty good, about 85% coverage of this pathway using these 3 different methods. Some specific examples, well, for some things, it really didn't matter how we enriched. So ERK1, ERK2, dually phosphorylated ACK loop sites, we see those no matter which enrichment we were doing, multi-pathway, PY, or IMAC. But for some things, the story was a little bit different. So mTOR, we identify a number of sites on mTOR, including important ones like 2448. You can see we only see this site in the multi-pathway. These are all serine sites, so we don't see them in tyrosine. IMAC identifies a couple of sites, but not all of the sites that you're getting with the multi-pathway. Same kind of story for ZAP70. So we see a number of sites, including the critical regulatory tyrosine 319. We see that site, though, just in the multi-pathway and PY, not in IMAC. And then one more example with P38, duly phosphorylated activation loop. Again, we see that with multi-pathway. We see it with phosphotyrosine, but not with the IMAC. So the IMAC does generate huge data sets. It gives you a ton of data from lower input material and is really a great kind of broad-based enrichment. But it's definitely not comprehensive. It's not giving you everything in the sample with that single enrichment. Now, what about quantitation in this experiment? Well, IMAC had the most peptides identified. It also has the most changes, both up and down, identified. But we do see changes quantitatively across all three of the different enrichments. And we actually followed up and validated some of these with Western. So for example, phosphozap70, tyrosine 319 phosphorylated, that went up about twofold in the mass spec data. We run the Western and we see a modest increase in signal in the CD3 stem cells. So nice validation there, consistent results there. PLC gamma, tyrosine 783. So that went up about 20-fold in the mass spec data. And we see a nice robust increase using that phosphospecific antibody in the Western blot. AKT actually didn't really change in the mass spec data, but again, that's consistent with our Western blotting results. And this is likely due to the fact that jerkats are P10 null, so they have high basal levels of AKT activation. And then ERK, duly phosphorylated at the activation loop residues, goes up about 32-fold in the mass spec data. And we go from not seeing signal to seeing a signal with that phosphoantibody in the Western blot. So in all cases, really nice validation by Western of the data that we're getting out of the mass spec study. So how much material do we need for all these different service options? It really runs the gamut from milligrams down to low amounts of micrograms. As I said before, when we're doing these motif or PTM antibody enrichments, we generally want to be in the milligram kind of range for protein. Same thing with the multi-pathway enrichment, although it somewhat depends on what PTM you want to look at. So something like phosphotyrosine, that's pretty low abundance. We really want to max out and get as much material as we possibly can. We can and we have done studies with less material, but your number of identifications starts dropping as you drop the sample amount. For other PTMs, it's not as critical. So we published a report about a year ago with a service client where they were only able to get us about 150 micrograms of material. We looked at acetylated lysine, and with acetylated lysine, we were still able to get over 1,000 ACK peptides identified and quantified per mass spec run, even from 150 micrograms of material. So it really kind of does depend on the enrichment that you're looking to do and the PTM you want to assay. For kinome view, we asked for about 1 milligram of material. For the IMAC, and this is another reason why IMAC has really been pretty popular, is we only asked for 500 micrograms of sample. So you get these huge data sets from less input material. As I mentioned, for the total proteomics, we ask for 100 micrograms or a couple million cells, but we can do projects with less than that when sample is limiting. And for a targeted assay, if you ever got to that point, it really depends on the endpoints you want to look at, but typically we're shooting for about 100 micrograms of input material for a targeted assay. So I've given you a sense of here are the different services that we do, here's how much material you need to do those. I wanted to finish off by just talking a little bit about how we kind of differentiate ourselves and our services and what's really important to us. And we took a lot of lessons that we've learned as an antibody company and applied those to our service business as well. And so it really all starts with us, with the quality of the data that we're providing. So we set the highest standards that we can for operation of our instrument. And this is some data from a recent ABRF study where they took the same sample and sent it out to a whole bunch of different mass spec labs. And each group of circles here is a different mass spec and a different mass spec lab. They're color coded by different instrument types. The y-axis is number of peptides identified. So they sent that sample out, you run it on your instrument, then you take those files and send it back to the study organizers so they search everything together. So the only variable here is operation of the instrument and type of instrument. And as you can see, CST came out on top. We had the highest number of identifications with that control sample of any lab in the test. So it really points to the care that we take in making sure our instruments are performing at their absolute best. We also are very concerned about data quality when it comes to quantification. So we talked a little bit about our ability to do the more accurate quantification of TMT total proteome data. But that carries over to PTM analyses as well. So when we're not doing TMT, this over here is data from a program called Skyline, which is available from University of Washington. And here we're looking at just 3 different samples. And when we're not doing TMT, we're actually directly measuring area under the peak, area under the curve for every peptide in every mass spec run that we do. And so anybody could do this. And I think what the difference with our services is that we get that data, now we generate fold changes, and then we take and we look at that data and say, okay, what things had a high fold change? Or what things had a high CV between technical replicates, replicate injections of the same sample? And we actually go back in and manually review that data to make sure that the correct peak was picked in every mass spec run. And when you do that, when you take that extra step, you get these data sets that are really nice, accurate, and reproducible. Reliable timelines we know is really important as well. So we have pre-study consultations, and whenever we have one of those, we talk about the turnaround time for any given project size. An enhanced data package. So when we started doing services, one thing that we knew we didn't want to be was the kind of place that just generated long lists of stuff and threw it at you and said, good luck, I hope you get some value out of it. So we've done things like making this PowerPoint that actually takes you through our tables in a lot of detail, going through column by column and explaining, here's what these numbers mean, here's how to interpret this data. Likewise, we also provide in our tables links to products for validation. So you'll notice some of these gene names, protein names, or sites are in blue. Those are actually hyperlinks. So in the gene name column, it goes to siRNA products. In the protein name column, it goes to total protein antibodies. And in the site number column, it goes to site-specific or phosphospecific antibodies. So right there telling you these are going to be the easiest things to follow up and validate because there are reagents directly available for them. And finally, these services are really consultative. So we have that teleconference or have that consult pre-study to talk about sample prep, sample requirements, even some of the finer points of the experimental design. We're always happy to jump in on that as well. As I mentioned before, after the study is done, we have that post-study consult to talk through the data, make sure everything is clear. And we're also not done once that post-study consult is done. We're available really whenever help is needed for emails, for a call, even doing things like helping to generate figures when that's necessary. You want to take a different kind of view of the data. And we've done these follow-ups 2, 3, even 4 years after a project's complete. So we really do believe in that, really think it's important to be available anytime help is needed. So that's kind of the whirlwind tour of what we're doing in my group and what we're doing in CST proteomics in general. Again, I think to me, the kind of most important things that we provide as a service provider are our quality, the experience we have, the speed of turnaround, and then the support you get. And this technology is really great for looking and finding biomarkers, determining mechanism of action, looking at cellular signaling pathways, and looking at proteins and PTMs. And so that's the end of my talk. And I think what we're going to do now is open it up to some question and answers. So I'm going to try and turn this webcam on.
If you put your questions in the questions tab, I will convey them to Matt. We have the first question is, I'm interested in both phosphoproteomics and monoubiquitination for signaling. Are the MS approaches much different?
Yeah, so they're actually pretty similar, and they're similar from both a sample prep perspective, where the methods there up front are very much the same, and running things on the instrument is basically the same as well. They don't require very different methods in running on the instrument. The big difference will be in how we search the data. So searching for phosphorylation as a variable modification versus that GG remnant as a variable modification. And one of the cool things actually is that when you're looking at different PTMs, like looking at phosphorylation and ubiquitination, you can actually use the same samples and immunoaffinity enrich those sequentially. So from the same sample, you don't have to prep twice as much material. You can actually do a phosphorylation enrichment and then a ubiquitination enrichment.
Thanks, man. We have oh, here we go. One more. Do you have general proteome data available for common cell lines? For example, if I was curious to know the expression of different protease classes in cancer cell lines.
So we don't currently have that available. We are constantly updating the data that we have in our Phosphocyte Plus database, which, despite the name, is actually a general repository for mass spec data on all of the different PTMs that we're looking at. And this is for internal work only, or things that have been published. But all the different PTMs, and we are starting to incorporate more of these large scale general proteomics studies. But that kind of data can be generated from any kind of sample if there's interest in looking at levels of different protease or other different protein classes across sample types.
Thanks, Matt. We have another question here. Are you expanding your PTM repertoire? Are there other PTMs you're working on for enrichment?
So there are. We do have antibody PTM and motif antibody development projects underway. One of the things that I would suggest is we do always like to hear from the customer about what they're looking to do, and that can actually help to influence development decisions that we're making, how we're prioritizing various projects. But yes, we are constantly developing new things. That we already have that long list that I showed in that slide, and that long list really continues to grow.
Here's another one for you, Matt. Is proteomics data easy to obtain from organoid tissues?
Yeah, absolutely. I mean, I think, the nice thing about proteomics is the flexibility as far as the type of sample that you can use. The biggest limitation generally is sample quantity. So it really depends on what you want to do. If you want to do a total proteome experiment, I mean, if we don't do labeling and multiplexing and fractionation, we can do those types of studies with, just a couple micrograms of material. If we have enough to see it in a protein assay, we have enough to run mass spec. Some of the other PTM stuff might be more difficult, just depending on the size of the organoids or how many you would need to get a certain protein amount. So something like tyrosine might be tough. But even PTMs like the IMAC enrichment or acetylysine, those would probably work well in the kind of protein scales that they're working in.
Thanks, Matt. We have another question here. When using multiple antibodies at once to multiplex the IP, what challenges have you seen? For example, limit on number of antibodies that can be used at the same time, or amount of antibody or solid phase matrix, etc.
Yeah, so that's a great question. So one thing is, we wouldn't mix antibodies from different PTMs just because then you have basically too complex a mix, if you're trying to do, for example, acetyl and methyl together. And then when you go to search that data and trying to match mass spec data with protein sequences, you're giving the program basically too many options to make a match between the mass spec data and the protein data. So the false discovery rates go through the roof. So we don't really do that for different PTMs, but when you're doing it with the same PTM, for example, those phosphoserine and threonine motif antibodies, there is definitely a trade-off. And so, what you will see is as you add in more and more of those motif antibodies to the mix, it starts looking more and more like an IMAC. where you get broader coverage, but you start to lose depth of coverage. So we don't typically really start seeing that until you get past, 3 or 4 different motif antibodies in the mix. But it definitely is an issue, and it is something to consider when you're designing a study. Should I do a single enrichment and again be a little more like IMAC and be a little broader, or should I really focus in on a given and more defined motif?
Thanks, Matt. Here's another one. Is there any available online database for diseases? For example, if we do proteomics on our sample, to be able to compare our results with data in the database?
Yeah, so I mean, there's a number of different databases online that talk about disease, talk about large proteomics data sets. One of the things that we have is the Phosphocyte Plus database, which again is a lot more than just phosphorylation. And we actually pull a lot of that accessory data from our data package right from Phosphocyte. And so one of the things that we include in that data package is actually disease associations. So for any given protein or site that's identified, we have there, if potential upstream kinase is known, downstream substrate is known, what disease associations there are to that protein or even to that particular site. So that is one great resource.
Thanks, Matt. Yeah, Phosphocyte Plus is something that I've also used quite a bit. We have another question here. Are there detailed methods available for publication, and where can the methods be found?
Yeah, so that's where having this kind of consultative service is really helpful. So for any given project, I have some kind of boilerplate methods, but I will usually take and go through and modify those for the specific project that is done to make sure that the methods are all correct for any publication. So that's something that I help out with, and then you just basically can copy and paste that right into your manuscript. The other thing that we do on the publication side of things is a lot of journals now are requiring the raw data files to be available. So we always keep those raw data files, and we have those available for you anytime you need them on request. And so you can upload those to a database. Now they're available, the journal is happy, the reviewers are happy, and you can go forward with the publication.
Just before I move on to the next question, I want to add in a comment based on our previous question about the database. One of the attendees, Marissa, mentioned that she finds the Human Protein Atlas helpful, but it's mostly based on RNA expression. So I just wanted to put that in there. Next question is, are all your PTM scan enrichment experiments done with label-free quantitation, or are you also using SciLac?
So we're doing everything on the PTM side label-free. And again, that's not any kind of just peptide counting thing. That's actually directly measuring the areas under the peaks for each of those peptides and manually reviewing those. And the reason we favor that approach over something like SciLac or reductive dimethylation is, so with those things, with the reductive dimethylation or with SciLac, you're limited in number of samples that you can have, just a couple of samples. The SciLac is a big pain. I did a lot of SciLac in the early days, and it's a big pain because you have to grow your cells in heavy and light media. Sometimes the cells don't like the heavy media. You have to go through a certain number of population doublings. You're in big trouble if you ever mixed up the heavy and light media one day, and now you'd have incomplete labeling. And then the other problem with SciLac and reductive dimethylation is that you're basically splitting up the signal. So one of the cool things that I talked about with TMT is that in the intact peptides, that data all stacks up as the same mass. Well, with the SciLac experiment, you have a heavy version and a light version of each peptide. And so you're basically now spending twice the instrument time to identify the same number of peptides. The instrument's going to spend time identifying the light version, and it's going to spend time identifying the heavy version. So you actually end up getting fewer identifications. And the way that we're doing the label-free quant actually ends up being really accurate. We are as accurate or more accurate than a SciLac experiment.
Thanks, Matt. Another question. When you analyze proteomics data, are there software programs to narrow down what you're looking for? For example, if I was interested in figuring out cell surface antigens specific to tumor tissue and wanted to screen for membrane-bound proteins, could I narrow my results to those types of hits, or do you have to navigate each large hit individually?
Yeah, so that's a great question. So actually, that's something else that is kind of in the data package already. So we have actually organized all the identifications we get by protein class, by GoSlim terms. And so there's a section for membrane proteins. There's a section for adapter proteins. There's a section for serine threonine kinases or tyrosine kinases or RTKs. And so all of that is in one sheet in the report, but it is organized by protein class, so that information is in there.
Just looking to see what the next question is. I also do want to mention that and highlight again the consultative approach that we have here. We realize that, especially with proteomics, one size does not fit all. So we really want to be able to talk with you about your project and understand the intricacies of what you're working for and be able to tell you, what is reasonable, what isn't reasonable, what you should expect to get out of it. So I just want to highlight that consultative approach that we have here. We want to partner with you, not just give you, as Matt said, a massive table with numbers and throw you to the wolves.
That's right. Stu, one thing I guess that I would add to that, since we've had a couple of questions about kind of the data and what comes along with the data package, is just to let everybody know that we have actually a full sample data package for what you would get if you did a service project with us. And so that can be made available to anybody that wants it. It's phosphotyrosine data, but kind of no matter what PTM you're looking at or even the total proteome stuff, the look and the content of the data package is going to be very much the same. So that can be a really useful tool in kind of seeing what the data package would look like if you did the service.
Thanks, Matt. I have another question here. Would the depth of IMAC improve and reach a level near that of PTM motif antibody if the input amount and enrichment scaled up? What service would you recommend for achieving both breadth and depth?
Yeah, so for the IMAC, more than I think increasing the sample amount, it might be a combination of increasing the sample amount and then fractionating those samples. The reason that we don't do that for the IMAC analysis is that I think we calculated out or tried to figure it out that it would be actually weeks of instrument time, which of course gets very expensive for a service, and you probably don't want to go through. And the gains are not exactly linear. So 14 days of instrument time doesn't give you as much, or doesn't give you 10 times as much information as 14 hours of instrument time or 1.4 hours of instrument time. So there is the potential to do that. And I think when you're talking about depth versus breadth of coverage, like I said, something that people do a lot is to kind of look at both sides of the coin. So they will do that IMAC to get a broad kind of overview, but they may do a more specific enrichment with a PTM or motif antibody. Most commonly we see that with doing the tyrosine, but there are other times where, for example, somebody might be interested in substrates of an AGC family kinase. And so they know roughly that the motif corresponds to some of those that we have in that list of serine threonine motif antibodies. And so they want to use that, one of those or a couple of those for the more specific enrichment, get depth of coverage. But in a lot of cases, excuse me, in a lot of cases, you may not be 100% that the motif is covered by one of those antibodies. And so they'll want to look outside that box as well. And that's when they'll do the IMAC enrichment to go broad and get more data. So, I think the best way to balance is you actually can do both.
Thanks, Matt. Another one here. How do you identify full-length novel proteins not listed in any data bank or paper from hypothetical proteins slash peptides?
Okay, good one. Yeah, so one of the nice things is that we have the ability to add basically whatever we want to the database that we use to match sequences to the actual mass spec data. So if there was a hypothetical protein that you wanted to add in to the database, we add that. And it's pretty cool. When we do the searching, we have, here are all the proteins, let's say human proteins, and then here is all the human proteins with reverse sequence. So we actually search the data with both forward and reverse proteins. The reverse ones we know are all wrong. They don't match to anything in real life. But that allows us to estimate false discovery rates. We see how many things are matching to reverse assignments that we know are wrong, how many things are matching after we've done our score filtering, and we can use that score filtering to set a target false discovery rate. So in this case, we might put in the forward and the reverse version of that hypothetical protein or those hypothetical proteins. And then if you have a match, you have that match at a given false discovery rate, and you have some confidence that it's not just kind of a random match between your hypothetical protein and the mass spec data.
Thanks, Matt. Another question here. How do we increase protein detection of hard to detect proteins in mass spec?
Yeah, okay, so always wanting to go deeper. I think there's a couple of things there. One, and I'm going to focus on the total proteome part of it, because with the PTMs, we gain depth and we gain specificity and simplify the mix of things we're delivering to the instrument through that enrichment. But with the total proteomics, I think the ability to multiplex and then fractionate has a huge effect on the depth of coverage. And so you're able to dig down and get a lot more proteins, thousands more proteins identified and quantified by doing that fractionation. We're also running long gradients on the instrument. So, over the years, we've basically figured out here is the longest gradient that we can run to really maximize identification rate without making the peaks get kind of broader and make the data a little bit messier. So those are really big factors for increasing depth of coverage. I would say it's running the instrument at the kind of the highest level possible, right? Again, we talked about kind of the strict QC metrics we have on our instrument that allows us to get the most out of every mass spec run, and then being able to fractionate samples to increase our depth of coverage.
Thanks, Matt. And we have one last question for you. When you have a PTM scan done, but there are no specific antibodies for validation, how do you ensure credibility and robustness?
Yeah, okay, so how do you validate this data? So, step one is, like I showed kind of at the end there, we have the products for validation right in the data package. And so the easiest things to validate are those where you have the specific antibody, right?
Yeah.
But as the question asks, what happens when you don't have that specific antibody? Well, so usually the kind of things that we recommend for that type of validation is there a total protein antibody available? Because then you can at least do a Western blot and see if there's a protein level change associated with the change you saw in the mass spec data. And if there is a total protein antibody available, you can do that total protein Western. You can also do IP Western. So let's say we were looking at phosphotyrosine data. If you have a total protein antibody, you could IP with the total protein antibody and then blot with the general phosphotyrosine antibody. And we have the same antibody available for Western blots that we use to actually do the enrichments for mass spec. And then you can do the reciprocal. So you can IP with the phosphotyrosine antibody, blot with the total protein antibody. And so we've done a lot, and others have done a lot of validation that way. I think there's also opportunity for mass spec based validation. So if something ends up being really the critical biomarker that you have discovered, or potential disease driver that you have discovered, that's when you might consider doing a targeted assay, or minimally having a heavy isotope synthetic peptide made and look at that in your samples to get absolute quantification.
Actually, I'm going to squeeze in just one more question, Matt, because we got one from one of the panelists. For phosphoprotein, site ID, and quantification between samples, how do you handle repeat proteins, e.g., the C-terminal domain of RNA polymerase II has around 53 very similar repeats that can be highly phosphorylated. Can these still be detected and quantified?
So they can be detected, they can be quantified. One of the things that we also include in the data is it's called a score or a mod score value, and that's a site localization score. So when you have one of these peptides that has multiple serines, threonines, tyrosines in it, and potentially very close to each other, that score tells you basically the probability that the program has put the phosphate on the correct residue, on the right serine, threonine, or tyrosine. And in some cases, you can actually get an A score that tells you this is confidently localized. In other cases, you may not, and that doesn't mean that the data is wrong. All it means is that there is still the possibility that this peptide is phosphorylated on this serine or this threonine that's right next door. So you can still get that data. You can still interpret that data. You just have to have a little bit of caution over the fact that if you don't have a high enough A score or site localization score, you're not going to know 100% if it's this serine or this threonine right next to it.
Thank you. And with that, we will wrap up the webinar. I want to thank you again, Matt, for fielding all the questions and the wonderful presentation. And I also want to just reiterate that this service is consultative. We want to work with you. So if you are all interested in further talking about this and looking at how this applies to your specific project, pharmacology group, discovery group, biomarker group, please reach out to your account manager and we would love to talk to you and understand what you're trying to accomplish and help you get there. So with that, I will end the webinar and thank you again, Matt, and thank you again, everybody.
All right, thank you, everybody.